All right. Anybody have any questions for me before we get started with all this? I'm just reminding everybody that this is our last lecture for Unit 2. So it's chapters 13 and 14 today. And then Thursday, we will have our presentations. So if you are a Unit 2 project person, Thursday, you will be presenting your project. And then next Tuesday is our Unit 2 exam, right? Everybody clear on that? We will have a review game session like we did last time. I will only be holding one, and it will be on Sunday, probably at like 6 or 7 on Sunday, somewhere around then. And it'll be like an hour, depending on how many people show up. So if I have like enough people and you guys want to keep going for an hour and a half, probably an hour and a half max. But it just kind of depends on how much, what you guys want to do. And it's going to be the same as last time. If you didn't join us last time, it's basically just playing Kahoot games like the ones that we play in the lab. Some of them will be team-based and some of them will be just basically answer the questions and go. But we will basically, I have like a set of 75 questions that it'll cycle through that'll help you guys basically practice with your knowledge on questions like that. Okay. So I'll send out an announcement on that review probably in the next couple of days. So you guys can have that in your schedule and know when be available for it. Doing 2 labs today in the labs also. Got some makeup to do with that. We probably, again, since we've got 2 chapters we're going over, we probably won't get to our questions today, but if we do, here's our victims. We're going to be doing 13 and 14 today. but the quiz will be over 10. Yeah. All right, we ready, folks? We're gonna get started. All right, reviewing chapter 10, which is videos that we should have watched by now. Hopefully you have. Which term refers to all of the chemical reactions that occur in a cell? All right, so all chemical reactions in the cell is going to be referred to, that's our metabolism. So your metabolism includes reactions that are anabolic and catabolic. So catabolic, like cats destroy things. Catabolic, that's going to be breaking down molecules to release energy. All right. And that's really what aerobic respiration is all about. What cellular respiration is another word for it. It's basically breaking down molecules and then harnessing that for cellular energy. All right. Anabolism, like anabolic steroids. Why do people take anabolic steroids? To build muscle, right? So building things is anabolism. Those two things together, and all of the reactions in the entire cell, that's your metabolism. Amphibolism is basically like utilizing, like breaking down things to get energy and utilizing intermediates from that and the energy from that to build things, like combining that together is like amphibolism. It refers to the interconnectedness of those two things. And the whole big old picture is metabolism, all right? All right, next question. Anabolism, I just said it, let's see if we're paying attention. All right, so this is going to be building molecules. So building is anabolism, catabolism is breaking apart. Next question. Enzymes are an example of what macromolecule? What kind of macromolecule are enzymes? We learned this in chapter 2. This still applies with chapter 10. They are proteins. Good job. All right, where does substrate bind to on an enzyme? The substrate binds to which site on the enzyme? All right, so the substrate is the molecule the enzyme is doing the work on, and that substrate binds to the active site. What are causes of a denatured protein? All right, so in order to denature proteins, a good portion of your protein structure is held together by hydrogen bonds and interactions between attractions within the protein. And so you can disrupt that by introducing too many extra hydrogen ions or maybe having too few hydrogen ions in the environment. So that's what your pH is going to be about. Or you can cause those weak associations to dissociate by adding in heat. Chemicals can also have the similar effect. We'll learn about how these things can destabilize protein and how that can be used basically to control microbial populations. When we get to chapter 11, which is our first chapter for unit 3. What best describes non-competitive inhibition? All right, so non-competitive inhibition. This is going to be referring, so we have two types of ways that you can inhibit a protein that's already made, an enzyme that's already functioning in the cell, there's two ways that you can inhibit it. And that could be competitive or non-competitive. If it's competitive, it's competing for the active site with, like, it competes against the substrate for the active site. So that inhibitor binds there first. Now the substrate can't bind there and can't have the action done on it to create the product, whatever that might be. So that's competitive. If it's non-competitive, then an inhibitor binds somewhere else, not the active site. Okay, and the two words for the site where it would bind, there's two words for it, and they mean the same thing. That's allosteric site or regulatory site. So that inhibitor binds not at the active site, the allosteric or the regulatory site. And that's where non-competitive inhibition will come into play. So it's not competing with the active site, with the substrate. Okay, so question 7, the 5-carbon sugar found in ATP. We actually introduced this in chapter 2 as well, but it became a big fixture for chapter 10 again. Creating cellular energy. All right, so the 5-carbon sugar that is found in ATP is ribose. If you guys actually had watched the lecture for chapter 10, then you would know that glucose has how many carbons in it? 6, and that's key for understanding how we get ATP from glucose. That's kind of hinting to me how many of you guys maybe haven't actually watched the lecture for chapter 10. Chapter 10, if you haven't looked into it, is a massively content-heavy chapter. So is chapter 6. If I had to tell you to focus on only 2 chapters for the whole unit, I would say 6 and 10. Do not skimp on 6 and 10, guys. And in fact, remember how in Unit 1, I told you that we'd have even numbers of questions for each of the chapters? Unit 2 exam is not like that. You're going to have more questions from the content-heavy chapters in this unit, okay? So bank on that, because these concepts aren't just big in my class. These concepts for genetics and then also enzymes and metabolism, you're going to see this again in physiology. If you haven't taken physiology yet, you're going to see it again. And these are going to pop up again and again, and they apply extremely heavily to the medical side of how our body works too. So understanding that. Also, for any of you who viewed my recording for this lecture, if you made it to the end and I had the relevancy content module, it was a video on COPD. If you don't understand how COPD ties into chapter 10, you're also not doing enough, okay? So be sure that you're actually making sure you're watching those videos and understanding that content and doing your part to understand all that stuff. So briefly before we go on, our 5-carbon sugar is ribose. So we learned in chapter 2 that ATP counts as one of our nucleotides. It fits in that like nucleic acid category box of our macromolecules. And our nucleic acids all consist of a nitrogenous base, a 5-carbon sugar, and a phosphate, right? And so this guy has all of that. So adenosine means we have adenine, which is like the GATC that we see in DNA, or the GAUC that we see in RNA. So the A is adenine. So what makes it adenosine is the adenine plus a sugar. And so the sugar in this case is ribose, not deoxyribose like we see in DNA, but ribose like we see in RNA. So that's a 5-carbon sugar. And then we have our phosphate. We happen to have 3 of them in ATP. And it's the fact that we have 3 phosphates covalently bound to one another. They have these strong negatively charged aspects to them. They're repelling each other, but they're covalently bonded to one another. So when we break those bonds between them, they are happy to leave, and the cell can use that energy that's released from that bond breaking to power things in the cell. So that's what the whole point is of ATP. All right, let's move on. Right, aerobic respiration requires what? Kinda in the name. I misspelled fluoride. I love it. All right, so aerobic respiration requires oxygen. I'm glad that we've got that at least. Don't forget that aerobic does mean aero, air, oxygen, okay? Because anaerobic, does it use oxygen? No, so that's the big difference there. This is something else. All right, gas produced by yeast during alcoholic fermentation. What gas do yeast make during alcoholic fermentation? Gas. All right, so the gas that's produced by yeast during alcoholic fermentation is carbon dioxide. It's actually how we get that fizziness in our beer. It comes from the fermentation. You can also infuse it with more carbon dioxide from, gas canister or whatever, but most of the time it's going to come naturally from the fermentation process. What happens with fermentation is you go through the 3 steps of aerobic respiration, right? Glycolysis, Krebs, and the electron transport chain. In fermentation, these are organisms that would love to go through all 3 of those steps, but if you don't give them oxygen, they can't go through the electron transport chain. And therefore, the whole point of Krebs is to collect electrons to put through the electron transport chain. So they don't need to be going through Krebs either. So they only go through glycolysis. And one of the products that's made from glycolysis, you get a little bit of ATP, and that's how they're surviving. It's getting their ATP that way. It's only a little bit. 2 net ATP just from glycolysis, whereas if you go through all of aerobic respiration, you get 38. So that's a big difference. So these guys are able to go through fermentation and get a little bit of ATP, but they've got pyruvate left over and they've got some electrons left over. You can't just have electrons collecting around. So they use those electrons, feed them back into pyruvate, and that's what makes our ethanol, our lactic acid, and our carbon dioxide, and whatever products can be made from fermentation that we like to use to make, bread and beer and wine and whatever else like that. So we are literally taking their oxygen away from them and forcing them to go through fermentation, which is just repeating glycolysis over and over again in order to get the products that we want from them. And that's the point of fermentation. Sticking them stuck in glycolysis. All right. During photosynthesis, plants use what gas to produce glucose as their food? So they're still going to use glucose in aerobic respiration. Plants still need glucose in order to make their ATP, but they do not get it from their environment. They have to make their own glucose to feed into aerobic respiration. So what molecule do they use to build that glucose? You get it from the air. Waiting on one person. Go. All right, so that's carbon dioxide. There's 2 steps to photosynthesis, the light reactions and the light independent reactions, or they're also called the Calvin cycle. You can call it the dark reactions too if you want to, that makes it easier for understanding, but that's not its official name, right? The dark reactions can occur during daylight. So that's why we don't like to call them the dark reactions. But the light reactions obviously have to happen in daylight. So the light dependent reactions, those light reactions, what happens is light comes in, it shines and hits pigments, and the pigments get excited and excite each other. And while they're exciting each other through that line in the membrane, protons are pumped out. So instead of, like we have with the electron transport chain, getting electrons from our food and feeding it through molecules and causing protons to pump out, these guys are getting excited by light only itself. And then using that light that's gotten excited to excite the pigments, and that allows the creation of our proton gradient that we can feed through ATP synthase, and that's going to create our ATP for us. So they create a little bit of ATP, but it's not as effective as using electrons to do it, but it makes a little bit, makes enough that's the whole point of the light reactions, taking light to make a little bit of ATP. Then the dark or Calvin cycle, the light independent reactions, they take the ATP from the light reactions and use that to fix carbon dioxide into 6-carbon glucose. They can use that glucose as an intermediate for building blocks for the whole cell, or they can use that glucose to feed it into aerobic respiration, because plants have mitochondria also. They don't just have chloroplasts. They also have mitochondria. And what happens in mitochondria? Aerobic respiration. So plants need oxygen too. So keep that in mind. All right, so carbon dioxide, we're going to fix that into our 6-carbon glucose molecules. And that happens in the Calvin cycle, photosynthesis. All right, great job, guys. Really did pretty good considering we didn't cover that in lecture itself. But if you haven't watched that lecture and taken your own notes on it, please do that, because remember, chapters 6 and 10 are pretty heavy on the exam. All right. Come on. Chapter 13. Chapter 13, we're going to talk about infection and disease. We know an infection is going to be any time that we have the establishment of microorganisms in the tissues or inside the host somewhere, and they're multiplying there. You can have an infection without having disease. So anytime that you have microbes growing somewhere where they normally shouldn't be, that's infection. Disease is anytime we have a deviation from health. So once we have a pathological state associated with it, so we start having signs and symptoms, that's disease. So you can have infection without disease. We've already talked about a third of the people with COVID don't have symptoms. Those people are infected, but they don't have disease, right? So they're able to act as carriers for that. We'll talk about what that is in a moment. Of course, we have normal biota within our body that has functions that are, mostly good for us. Our indigenous normal flora that can include bacteria, fungi. We know we can include bacteria. We're aware of the gut bacteria, right? It also includes fungi. So mostly is talking about yeast here. Okay, so like yeast, that's part of the normal skin biota, as well as like the genital tract. We're aware of having some yeast in your genital tract, normal, 100% normal. It just shouldn't be overgrowing, right? And so having your populations of your normal biota in the normal ratios and numbers, that's your microbiome. So bacteria, fungi, mostly yeast. Sometimes protozoa can be part of your biome, and viruses are heavily part of your biome as well. It's very, very understudied right now, but they are. The only group that is not part of your biome is going to be the helminths. You shouldn't have any helminths in your body at all. All right. So here we have a slide basically describing to us, oh, whoops. Why is this not? This toolbar here. I can't pull up my, there it is. So here we have humans being colonized by microbes. Some of these guys are your resident microbes. You might have a few that could be potential pathogens that may be either introduced into your body by, like, let's say I pick up my phone right here. Is my phone sterile, guys? No. If I put my phone up to my head, am I going to get an infection and a disease from it? Probably not, right? But am I introducing those microbes to my body? Yeah. So those usually are going to be transient microbes. We come into contact with plenty of transient microbes in our life that aren't going to be causing disease. Either there's just not enough to cause infection, or your body's able to fight them off with your immune system, right? So typically, if you have a healthy immune system, those sorts of kind of interactions with transient microbes that could be potential pathogens, your body can get rid of it pretty easily. So you have these microbes that are going to be resident microbes, or potential pathogens, or transient microbes, things that you'll come into contact with and your body can just still exit, and there's no issue there, right? All right, so that would be great, then they're gone. So that's a possibility too. So we can have potential pathogens that are part of our resident biota, or we can get introduced to potential pathogens outside of our biome. So if you guys have COVID and you come in here and I get infected from that, obviously that came from outside of my biome, and I can get COVID from that. Or I might have E. coli in my colon, and then I, practice poor hygiene, or I have a sexual encounter, and that bacteria gets introduced into my urinary tract from my colon. And that's might lead to an infection in my urinary tract from bacteria that doesn't belong there. But it came from within me. But still, that's an infection. That E. coli doesn't belong in my urinary tract and will establish an infection and potentially disease. You might have that burning when you urinate, the pain, the constant urgency, and maybe blood or pus in the urine. Yes.
So once you have like symptoms,
that's disease. Yes. Mm-hmm. And so it could be symptoms that you would recognize. But we know there's even patients out there, far too many anymore nowadays, who have just gotten so used to having to deal with an infection that they might not recognize that they're having signs or symptoms, that you as a healthcare worker would be like, yeah, but you're not supposed to have pus coming out of this orifice of your body or something like that, right? And so signs and symptoms, typically they shouldn't be too subjective, right? If you're having a deviation from your normal health, but sometimes chronic symptoms can be just seen as part of your everyday life and unfortunately aren't recognized as signs or symptoms anymore. So that was a good question. All right. So from outside or from inside your body, going to an area where it shouldn't, overgrowing in areas where it shouldn't. those are going to lead to infection. And sometimes infection might get cleared before you have symptoms. So people who are walking around with COVID that are asymptomatic might just clear that infection and never have symptoms. And that would just be that situation. But if it leads to disease, once you have your signs or your symptoms or what you perceive to be deviation from health as a result of that infection, now you have disease. Sometimes when you clear that disease, it can become part of your biome. And you plus your biome, the term for that is the holobiont, right? So you plus your biome. It's not just you living in this body, right? It's also all the different microbes that are living in here too. And that's your holobiont. So once you have a disease, you might kick it, but it could potentially, in some cases, become part of your biome. This is not uncommon when we see like staph infections or C. diff, for example. If somebody gets C. diff, even if we are able to clear it, there might still be pockets of it in their GI tract, and it could become part of their normal biome, where it's not creating disease necessarily anymore, but it's still hiding out in their gut as part of their biome. And so at that point, it could become part of that. You get a staph infection in your skin, it doesn't always have to be MRSA, right? Staph's everywhere, but it could be MRSA. You could get that staph infection, get it cleared up with some antibiotics, but maybe not cleared all of it, and it could become part of your biome. You know, people do carry staph as part of their normal biome, about 4% of the population, and a lot of people got that from somebody else. So, yeah. Also, when you were growing in the womb inside your mom, which is one of the things we're about to talk about here in a second, as you're growing in the womb, that's, I don't want to say sterile because it's not completely sterile, but you're not exposed to very many bacteria in there. It's not like you're born colonized with bacteria. So where does the bacteria that's on your skin and in your gut and in your throat and your sinuses, all of the parts that make up what we consider your microbiome, where does that stuff come from if it didn't come from birth, right? Or maybe it came from the birthing process, but not from the uterus itself, right? Because the cells that you're building when you're dividing and becoming an organism in the womb don't include your biome, right? So, all right, anyways, sometimes disease, we know it can lead to death. So let's not forget that. All right, Human Microbiome Project is a project basically to try to study what organisms we might commonly find in what areas of the body. Our biome, it obviously serves a purpose for us. It's easy enough for us to think about how our gut biome is really important for helping us digest our food. The bacteria that live in your gut, and we're talking about millions in each gram of stuff in your gut, that those guys are there to break down your food, and then their products that they make from breaking it down, you can use as nutrients. So it's a symbiotic relationship because they're depending on us to give them shelter and food, and then they are breaking things down to create products that we can then use for food as well. So we're working together in that, and our lives are depending on each other for that. So we have them essentially being able to create nutrients for us that we can't do for ourselves. They will help train our immune system as to what is good or what's bad or what we should react to and what we shouldn't. They'll help in the development of our organs, how our body basically reacts to our microbiome in certain areas can help with making sure that our organs are functioning optimally the way that they should, especially in our gut. And there's something called microbial antagonism. So there's a few ways that microbial antagonism works. The first example is this stadium seat example that I like to give. So you have a big old stadium that's full of a whole bunch of seats. You go to Boone Pickens Stadium, right? Whole bunch of seats there. Your microbes should be taking up most of those seats, especially in your gut. We have good guys that are taking up the seats so that bad guys can't sit down. So think of it that way. So they are filling up like basically spaces, literal physical space, as well as using nutrients in those areas and things like that. So the bad guys come in and they're like, there aren't nutrients available. There's nowhere for me to adhere to. There's nothing that I can do with this. So that's one way that microbial antagonism works. Another way that microbial antagonism works is that our microbes are like maybe creating toxins or other antimicrobial molecules that will fight off the bad guys that could come in. So literally fighting against them with their weapons or just taking up the stadium seats, right? So that's how microbial antagonism works. Microbial antagonism is our biome basically making it less hospitable to bad guys, like pathogens. Most of us are aware of different factors that can increase your susceptibility to infection. We're not going to go over these in detail or anything, but age, pregnancy, and things like this can put stress on your body and make your immune system weaker, and also could potentially make your microbial antagonism of your biome weaker as well. It just depends on what your body's going through. Any infection caused by the biota that's already in your body is called an endogenous infection. So we have endogenous infections and exogenous infections. So endogenous infections like the E. coli from my own colon getting into my, UTI situation, that would be an endogenous infection. My E. coli belongs in my colon. It doesn't belong in my urinary tract. 80 to 90% of all urinary tract infections in women are caused by the E. coli in your own colon. So that's why we advocate wiping from front to back. That's why that exists. That's a hygiene issue. And then urinating after sex. You got a whole lot of mashing around going on in that area. So you want to be sure that you're flushing everything out where you can so that these E. coli can't adhere and then lead to the development of a urinary tract infection. So that's E. coli from your own bottom that gets into the urinary tract most of the time. Okay. So that's an endogenous infection. Another thing you can have is, let's say that you get the flu. That's exogenous, right? You don't usually have that as part of your biome. So you get the flu, that's exogenous from somebody else, but now your lungs are getting damaged from the flu, your respiratory tract's getting damaged from the flu, and the normal biome that's in your sinus cavity where it belongs, just in small numbers, including Streptococcus pneumoniae, the number one cause of community-acquired pneumonia as well as meningitis. Okay, normal lives in your sinus cavity, but now you have the flu, it's damaged your lungs, and this guy found some stadium seats because the normal biome and the normal way that your lungs are functioning, we've created stadium seats or we've opened up stadium seats. Now this guy can move out for this opportunity to move into a new part of your body and grow in an area where it shouldn't be growing, and then lead to the development of a secondary infection of pneumonia as a result. These are endogenous infections, okay, from your own body. So where do we get our biome that's going to be taking up our stadium seats for us? So we think there's some colonization that just happens from being in the uterus. It isn't a sterile environment. So you will have some introduction from the mother's biome just in her uterus. But most of your biome is going to get introduced through the steps that come after that. So the first biggest exposure that you're going to have is through the birth canal. The birth canal has its own biome, and these are going to get introduced through the eyes and the mouth of the baby as it's being born and get introduced into that baby's biome. It's going to help colonize the gut, the sinuses, the throat, and the skin, and all of these areas where we have our biome that acts as, microbial antagonism. If you're born via C-section, you don't get exposed to this, right? So you're missing out on your opportunity to create your biome there. Additionally, another place that we get a very large part of our gut biome in early ages as infancy is through breast milk. So if you're not getting exposed to breast milk, you're not getting exposed to the microbiome that's contained within the breast milk. This includes bacteria that can break down the milk effectively and the proteins and sugars that are in the milk that your gut bacteria, that your gut by itself without the bacteria wouldn't be able to break down. And so if you have a baby that's born by a C-section that does only get formula that doesn't have any probiotics in it or anything, so that baby's a lot more colicky and then have a lot more GI issues earlier on in life until it gets a good biome built up. So these are things that I feel like are pretty underappreciated in the world of medicine that maybe doctors and nurses are aware of, but the patients aren't educated enough on in order to understand why their babies are having these issues. And so they want to blame it on a disease or something like that, when it's probably just a lack of a microbiome that's doing usually work for the baby outside of that. So if you are somebody, it's perfectly fine to have a baby by a C-section and to feed it formula, but be sure that you or your family members or your patients are feeding the baby formula that has probiotics in it. So these are bacteria that are going to be good and beneficial to the gut of the baby while it's developing. Really important to get that. The next place that we get our colonization from is, of course, your caregivers and your family, and then your environment. So getting little licks on your face from your doggy, or you're petting your kitty, you're going outside and playing in the grass or whatever, you're getting colonized by bacteria. And does your microbiome stop there? Of course not. So as you are aging, especially once we hit puberty and the pH and the hormones in your body are changing your body, your microbiome will adjust to that. As we're getting exposed to sexual partners, our microbiome will adjust to that and incorporate some of their microbiome with ours and vice versa. So your microbiome is constantly changing and developing in accordance with your environment. All right, so we have talked previously about what a pathogen is. A pathogen is any microbe that will lead to infection and disease, potentially. So if it has the potential to lead to infection and disease, that would be a pathogen. We have true pathogens and opportunistic pathogens. A true pathogens, basically these are disease causers. you're going to cause disease even in healthy people. So this is COVID, this is flu, this is stuff that's not normally part of your biome, but that you get exposed to. Tuberculosis. You don't have this just hanging around and then it goes somewhere else in your body and grows up. It's these get in your body, they cause disease. So that's a true pathogen. They are disease causers. That's what they do. Opportunistic pathogens. These are guys that are either normal in your body. They could be normal in your biome, or they're normal in somebody else's biome, or there's something in the environment that normally doesn't cause disease. Right now, as we're breathing in the air that's around us, do we think that that air is sterile? It better not be thinking it's sterile, because it isn't. So we have plenty of mold and bacteria and spores and things that are hanging out in the air. So you might be breathing in right now a mold called Aspergillus fumigatus. Really, really, really common. It's just all over the place, right? And your body should be able to deal with that just fine. It won't cause any disease, and it's not trying to. It's just being a mold, doing its mold thing. But sometimes if you have a sinus infection, we've mentioned this previously, you have a sinus infection, it will wall off a nice little warm, moist area for a couple of those mold spores, and then they'll be able to grow up in your sinuses. That's an opportunistic infection. It doesn't normally cause infection. It's not its goal in life to exist as a pathogen, but you created a situation where it can take that opportunity to be an infective organism, right? So then it will be a pathogen. So those are opportunistic pathogens, things that normally don't cause disease. That's an example of an exogenous guy. But just like we talked about with E. coli belongs in your colon, right? It gets introduced into your urethra, into your urinary tract. It's going to take that opportunity to become a pathogen by creating an infection and disease in your urinary tract, right? Right, so virulence, we've kind of mentioned this term a little bit, but pathogenicity is referring to the fact that you're causing a disease. So you're having a deviation from health. That's pathogenicity. The degree of pathogenicity, how bad are you causing the disease? That's virulence. So anything that helps an organism be virulent is a virulence factor. So having a capsule helps you evade the immune system, virulence factor. You know, secreting toxins that cause damage to your host, virulence factor. So these are pretty obvious things, but anything that you could envision as helping that organism cause disease in the host, not just helping it multiply. You know, being able to multiply isn't a virulence factor, but anything that helps it cause disease. So having signs and symptoms, damage to the host, those are things that will be virulence factors, okay? This brings us also to the infectious dose, the minimum amount of organism that you need to cause disease. So this is different for each pathogen. So if we talk about a lot of our viral diseases, those might only need one virus on average to get into the body, maybe 1 to 5 viruses, viral particles. Otherwise, they could be just transient organisms. When we talk about salmonella, for example, it requires about, on average, 50 bacteria in order to cause disease. So you have 1 or 2 salmonella bacteria introduced in from eating your, over easy eggs. I like a runny egg yolk, y'all. I love a runny egg yolk. So you could potentially be introducing some salmonella by having your runny egg yolks, but it might only be like 1 or 2 of those bacteria. And that shouldn't typically, in the average person, cause disease. It'll be transient. Your body will get rid of it and you'll be fine. But in order to cause an infection, we would need at least 50 of those salmonella. And once you introduce 50 of them, then maybe you're going to be causing an infection. And then maybe that infection will lead to disease. Okay. All right, so here are the steps involved in infection, and they're pretty straightforward and easy to understand. I'm going to go over them briefly. The slides that follow go in a little bit more detail. We'll just touch on some of the detail as needed. But getting in is the first step. That's finding the portal of entry. Broken skin, getting in by being eaten and getting through the GI tract. You got to survive the stomach if you're going that way, right? Things like this, getting breathed in through the respiratory tract, getting in through the urogenital tract, and all of these things. Or it could already be endogenous biome. Maybe it's just overgrowing. Attaching and negotiating the microbiome. So getting a spot, so attaching somehow. Establishing yourself somewhere. And so this is finding a seat in the stadium seats, right? Or it's like literally just physically adhering somewhere in the host. So we know with our microbes that we talked about previously that bacteria have 2 ways that they can kind of adhere. They have their fimbriae, those are attachment kind of appendages that they have, fimbriae, or they could have capsules, those or slime layers. So those capsules or slime layers, that glycocalyx, that sticky coating can allow for adherence as well. Our viruses have the spikes that allow them to dock to those specific doors, basically, to get in to the cells. All right. And surviving our host defenses. This mostly means avoiding or surviving phagocytosis. This is our white blood cells eating them up. They've got to either be able to avoid this or survive it. We know capsules help with avoiding it. There are things like Mycobacterium tuberculosis that causes tuberculosis. It gets eaten up by the white blood cells in your lungs. They're alveolar macrophages. These alveolar macrophages, the alveoli are the little sacs in your lungs. Macrophages are the type of white blood cell they are. Come up and eat up the Mycobacterium tuberculosis bacteria, and they want to be there. That's where they live. Yes, they love hanging out in those macrophages. So your body's just trying to defend itself. They come to eat up those guys. It's what they're for. It's their job. And then these guys get eaten up, and they're like, "Thank you so much for creating me my brand new home. I've been looking for a new home for a while," and they survive there. So they have mechanisms that allow them to be eaten up, but they won't get killed by our macrophages. So there's that sort of stuff as well as maybe mimicking your host molecules. So things like strep throat, Streptococcus pyogenes, covers itself with proteins that look like our proteins. They're very similar, and your body might not be able to recognize it as foreign. If your body does recognize it as foreign, it'll look so similar to self that you end up attacking self, and you can actually develop temporary autoimmune effect because of this. So while you're infected with strep throat, you could also be attacking your body. This is actually what leads to the development of rheumatic fever as a result of strep throat, scarlet fever as well. All right, then we have damage caused by these guys. This is really what we're talking about, is the development of signs and symptoms. So anytime we're causing any damage to the host, this is oftentimes going to be led by your immune response. So whether you have a fever, microbes aren't forcing you to have a fever. Your immune system sees microbes and raises your temperature in your body. There's reasons for fevers that we're going to talk about in chapter 18. That's a unit 3 thing. We'll talk about fevers and inflammation and what the whole point is of fevers and inflammation. But hey, I just want to give a little heads up out there. The fever has a purpose. It does work for your body. It helps fight the microbes. You get a fever and it's within reasonable, manageable range. You don't need to be treating it.
Not 105.
Not 105. Yes, please don't wait till you get to 104. Probably once you're hitting 103, you might want to start taking that Tylenol, right? And of course, if you have little kids, it's a different situation. Please consult your doctors. But as adults, as long as you're not like severely uncomfortable in pain, let your fever run its course. It has a purpose, okay? So we'll talk about that in chapter 18 though. But anytime you're having damage being caused to the host by this infection, that's disease that's going to be developing, usually as a result from the damage being caused by the microbe or the damage being caused by your own immune system trying to fight off those microbes. Okay. Then we have getting out. That's the exiting the host. You can basically be creating exudate that could include pus, or it could be these microbes leaving your body through urination or defecation, coughing, sneezing, all of these things. So we're going to talk on some of this stuff. Getting in, we've already mentioned that infections can be endogenous or exogenous. Exogenous from outside the body, endogenous from in the body. Okay. We can also have microbes that can cross the placenta. We usually think of the placenta as being a barrier, but for some microbes, they've developed in a way that they can actually use this as a highway mechanism to get from the mother to the baby. This is not as common as it is for this to actually be a barrier. It's actually more common. It works more commonly as a barrier, but some diseases are able to cross this. All right, becoming established, we've talked about adhesion. Don't forget about quorum sensing, which is whenever our organisms that exist in a biofilm can kind of read the room, if you will, from waste products and signaling products made by the nearby bacteria. We talked about the fimbriae, our attachment appendages. Then we have capsules, our sticky coating. So these are our bacteria. And our protozoa can do these as well. And then down here we have our spikes for our viruses. The spikes that will basically dock with their appropriate docking molecule as like the key into the lock sort of situation. All right, then we have to survive our phagocytes, our white blood cells that are constantly in our immune system going around trying to eat up any waste or foreign material and digest it down. Anything that helps an organism avoid either being phagocytosed or being destroyed after phagocytosis, these are called anti-phagocytic factors. Anything that an organism has that is an anti-phagocytic factor is also a virulence factor, okay? So anti-phagocytic factors are virulence factors that help either avoid the phagocytes or avoid being digested by the phagocytes, or it might be that they kill the phagocytes once they get in there. So other things that could be considered virulence factors could be structures like flagella that help them get around to different areas in the body. Having fimbriae. So we know that E. coli that cause your urinary tract infections actually have stronger binding attachment fimbriae than like what typical bacteria would normally have. That allows them to kind of survive the flushing mechanism of the urine. That's a virulence factor. And other adaptations like toxins, secretion of toxins and things like that. How do we cause damage to the host? So this is going to get to talking about some of those toxins and enzymes that these organisms can make. Some of our bacteria make enzymes that help them break into deeper parts of your tissues. So we have different kinds of enzymes, such as exoenzymes. So these enzymes, again, proteins, and their names are going to end in ase. So we have some examples down here. We've kind of introduced another one that was called collagenase. So ACE enzyme, what do you think it does? It breaks down collagen. So collagen is one of your extracellular matrix proteins. And so these guys have an enzyme that can break that down that helps them get into deeper tissues. Hyaluronidase, same thing. Hyaluronic acid is part of your extracellular matrix. It helps maintain the moisture environment outside of your cells. So it'll disrupt your moisture maintenance environment as well as allow it to break in deeper into your tissues. Mucinase, excuse me, mucinase. So this is an enzyme that will break down mucin, which is what makes up your mucus. Mucus is there to be thick and sticky to trap microbes so it can't get to the tissues of your respiratory tract or your intestines or things like that, right? So whenever we have mucinase, this is an enzyme that will break down that mucus and make it so they can get into those deeper tissues even better. So all of these defensive mechanisms that we have in place in our body to trap these guys, there's usually a guy there that can hijack that situation and break it down somehow and end up saying, you know what, you tried, good job, but I'm gonna win this one, which is how we. Yeah, yes, that could potentially cause runny noses if you were talking about an organism that can make mucinase. So I want to be very clear that viruses don't make mucinase. They don't have all of these genes in them to create all these different kinds of enzymes that are like that. But bacteria do. So if you had like a bacterial sinus infection and you're having a runny nose, that could be from the inflammatory response trying to flush things out, but it also could be from mucinase. So breaking down that mucus and then allowing it to get in deeper. Viruses like influenza. Influenza has something on its surface called neuraminidase. So H1N1. We know that about flu, right? HN. So that's hemagglutinin and neuraminidase. So hemagglutinin is how it binds to our cells, but neuraminidase has a few functions. It allows that flu virus to basically break into our cells, to bud out of our cells, but it also helps breaking down mucus. So it kind of carries around an enzyme on its surface. It doesn't produce it as an exoenzyme, it doesn't secrete it, but it has it on its surface. It helps break down the mucus in your lungs. So it is kind of adapted to be able to do that. But again, that normally wouldn't happen in like your sinus cavity. That would lead to a runny nose. It would happen in your lungs themselves. But yes, so they have those sorts of similar kind of things that they could do. But yeah, there's a whole slew of other ones. These are just examples. Anything that causes damage to the host that these guys will produce are called toxins. So we have two forms of toxins, exotoxins and endotoxin. Endotoxin is always LPS. And that's always in gram negatives. Endotoxin, that is what endotoxin is, period. Okay. Endotoxin is lipopolysaccharide, which is that molecule on the outer membrane of our gram negatives. So I told you guys we were going to be talking about this stuff a lot, and we are. We ain't done with it, and we're not going to be done with it. So if you're gram negative and you have that outer membrane and you have lipopolysaccharide just as part of it, that's seen as a toxin to our body, and it's considered an endotoxin because it's part of the bacteria. It's the only endotoxin that there is. So if you're ever encountering somebody going into toxic shock caused by gram negative bacteria, then that's usually going to be caused by the presence of endotoxin. Our body sees it as foreign immediately, and will go into usually a shock type response in response to the presence of LPS. Okay. So why doesn't our body attack the gram negative bacteria in your gut? Talk about that. Okay. But if it gets into your bloodstream, it's bad. Okay. All right, so exotoxins, these are obviously things that are going to be exo outside of our bacterial cells. They're secreting these guys. So bacteria will secrete toxins. They aren't always enzymes. So anything that they secrete that can cause damage to the host, that's going to be an exotoxin. If it's an enzyme that also does that, well, enzymes can be exotoxins too, but they'll also be exoenzymes, right? So an exoenzyme can be an exotoxin that's causing damage. All right, so example of this would be like a botulism toxin. All right, so that would cause paralysis of our cells. It's not an enzyme that does that, but it is a toxin that has that paralyzing effect on our cells. It's an exotoxin. Hemolysis is a good example of this sort of effect. So we're talking about enzymes that break down red blood cells. So we learned about this earlier on, that we have gamma hemolysis, alpha hemolysis, and beta hemolysis. Gamma hemolysis, we have growth, but no actual breakdown of blood cells. No blood cell breakdown. Alpha, you have partial blood cell breakdown. So you'll have a greenish tinge. And then in beta, this one was gamma. In beta, we have total breakdown of blood cells. And what we'll see as a result of that is clear area around it. All right, we talked about this previously. It won't be the last time for that either. So hemolysis is hemolysis. Hemo is blood, lysis is breakdown. All right, moving on to excessive host response. Some microbes can actually cause your body to overreact to them. They might have molecules on them that your body just, like endotoxin, right? So our body can have an excessive response to that. That's probably maybe stronger than necessary in order to deal with the infection. And then that can lead to development of disease response to that thing as well. Another example that we have given in the slide is with COVID-19. When you get long COVID, it's almost always caused by that person's over response to that virus. That person's body was overreacting. Their immune system was overreacting to that virus being there. And then you will have symptoms and signs persist for months, even maybe after clearing the virus. There's different patterns of infection. We have localized infections, which are exactly what they sound like. This would be like a boil. You have an infection at one spot in the body, or systemic infection. This would be infections that are all throughout the body. We have focal infections. This is where you have one site of infection that moves to another, but it's not all over the body. It's just moved to another spot, right? We have mixed infections where you have more than one microbe involved. And then we can have primary and secondary infections. You had a primary infection, maybe you treated that, and now because you treated that, you're going to get a secondary infection. Or like we talked about with the flu, when you get the flu, you get a virus in your lungs that's causing damage in your lungs, and now other bacteria are taking up the stadium seats that it's creating or that it's freed up because of causing damage in that area, right? So that's a secondary infection. You get a UTI, you go take an antibiotic, it can clear out all the bacteria that's in your reproductive area, in your genital area, and then now you killed off the bacteria, but what's left? Yeast. Yeast aren't killed by antibiotics. They're not bacteria, they're fungi. They're killed by antifungals. So the yeast will overgrow because you just cleared out all your stadium seats with your antibiotic. So now you get a yeast infection. Or because we have a weird immune response going on to a yeast infection that could be developed from having that antibiotic, you take your antifungal and I cleared out the fungus, but now these other bacteria are growing up. They're in weird numbers compared to what they're supposed to be. You're supposed to have a whole lot of lactobacillus and a whole lot of these other things, but maybe other things are growing up instead, and now you've got BV. So it's bacterial vaginosis. It's when you have bacteria growing in the vagina area that is not supposed to be growing in those numbers. They're normal for the area, they're just out of proportion. So that's BV. Okay, so all these can happen just from disrupting the flora in the area. Yes. So mixed infection is caused by various microbes. That's what this one is. Like just a number of microbes. So we have infections that can, maybe skin infections, are not usually just caused by one microbe. There could be any number of microbes that may have been introduced from the environment or the skin or something like that, right? So usually what is happening, something like that, is they have it as part of their normal microbiome, and they are repeatedly putting themselves in a situation where those stadium seats aren't enough. So it's because we still have some of the bad guys in the stadium seats, right? So the bad guys are still there. So anytime that we have even a slight shift in an opening of a few stadium seats, those bad guys are going to try to overpopulate and take that up. So they are used to that. They become adapted to that and that sort of stuff. So people, we often see this with strep. Strep is a really common one for that. And so is staph. Staph is pretty common for that as well. So these guys are basically fine-tuned to overgrow if they get an opportunity. Yes.
Okay, so like with autoimmune diseases, what like specifies it? I don't know. Like,
We're going to talk about that in chapter 17. Okay. 17, like a good portion of that is.
So with the primary and secondary, are you talking about like if you have the flu and then that flu, you also
yep. Yes. Right. So flu itself can cause pneumonia. It's not actually that common for it to do that. So a lot of times what we see with flu is you get the flu and then you get pneumonia caused by bacterial infection. And so because you have your damage being caused by the flu virus or your immune system trying to get rid of it, those things going on in your lungs, that other bacteria from your sinus cavity will be able to take hold in there, like what we talked about earlier with the Streptococcus pneumoniae. So that's actually pretty common when you get the flu, the damage being caused by it, and developing that secondary infection from another bacteria. It's a separate thing. Yeah. Make sense? Okay. All good questions. And we'll talk about some of these specific diseases when we get into Unit 4, right? All right, so we already kind of went through these slides, so I'm going to look into more detail on these. You are welcome to. Acute versus chronic. An acute infection is something that will come on rapidly and usually have short-lived effects, but we have usually stronger signs and symptoms up front. Whereas chronic infections come on gradually and last much longer, but often also have a more muted signs and symptoms as well. Whether something is chronic or acute, it depends on the disease that you're talking about and how it's being presented. So we can't just say just because you've had symptoms for 2 weeks that makes it chronic automatically. It just depends on the disease that we're talking about. A sign, a symptom, and a syndrome. A sign, I literally, when I think of signs versus symptoms, so think of those two as being with each other. Oh my gosh, why? All right, sign. I literally think of a stop sign when I think of signs, okay? If I had a stop sign here in this room, could you touch it? Like literally, yes. Okay, it's a physical object, right? You can verify that it is there. You can't say the same thing of the word symptom. It's like a little bit more amorphous. And that's exactly how the definitions for these guys work. Signs are things that you can verify. You can measure them. You can observe them. You can observe, exactly. You can observe vomiting. You can observe a fever. You can measure it. You can observe swollen lymph nodes. You can palpate them. You can observe that they have a high white blood cell count, or that they have low iron, or that they have diarrhea. Can you observe or measure pain? No. Can you observe or measure fatigue? No. Can you observe or measure nausea? No. So those are symptoms. These are subjective. Yes, signs are objective. You can measure them. They are verifiable. Yes, so at face value, right? And then there are some tests that you can do to test like muscle strength. So when they're telling you that, oh, I've just been so weak lately, like my muscles just have been so weak. Well, it's a symptom until you put into practice. Maybe there could be tests that you could utilize for that. But like most of the time, if you're coming, if a patient comes into you and they're saying that like they've had a cold, oh, my body's just been so weak, I just can't like function, then that's a symptom, right? We're not usually going to be measuring muscle strength and weakness and stuff like that for that. But if they're having something like, let's say like they're having some sort of, I don't know what a good example would be, like MS or something like that. They're talking about muscle weakness. Then you would probably be using like some ways to measure their muscle strength or weakness. So we're talking about with like veterinary care. Those are usually going to be measured by the vet, right? By looking at the dog's joints and how the dogs will be able to walk, will be able to support their weight. That's a little bit more measurable with an animal actually than it is with people, where they're just reporting these things. It's definitely a much more difficult thing with pets because they can't tell you that they're in pain. And pets often will be in pain and not have any sign that at all to the owners that they're in any pain at all. Right. And they'll just like be putting up with it. And they might just seem like tired. Well, he's been so tired. Well, what's actually by making him act like he's tired? He's in pain and he just doesn't want to do as much, right? It was one of the compounds where like the bone was literally out of the skin. So yeah. Completely different than people, right? Well, people are going to let you know they stubbed their toe, right? So anyways, so these are things that it's definitely going to be different if you're dealing in the veterinary field as compared to like human medicine. But with human medicine, a lot of these symptoms, these are things that your patients are telling you about, and you just have to take it on faith that they're experiencing. Even when somebody's having a migraine, you can't actually verify that they're having that pain. So it's an unfortunate thing, but that's still a symptom. Doesn't make that they're not real. They're just, we can't measure them and verify them. Okay? We can't quantify it. All right, so a syndrome. What's a syndrome? A syndrome is any sort of disease that is defined by the signs and symptoms, not by the cause. And so this is actually pretty easy to give you guys an example of. The flu. What's it caused by? Influenza virus. Cold. What's it caused by? A lot of different things can cause the cold, right? So adenovirus, rhinovirus, RSV, all of these different viruses. There's over 200 different viruses that can cause the common cold. So what's a common cold? Well, having a runny nose, maybe some pain in the sinus cavities, sore throat possibly can be associated with that. Usually not a fever, or if there is a fever, it's very low grade. And we don't have the muscle weakness and the muscle pain and the tiredness, the fatigue that's usually associated with the flu. So common cold is usually a syndrome. It's defined by the signs and symptoms, not by the cause. Meningitis is another good example. Meningitis is defined by having that photophobia, which is light sensitivity, stiffness in the neck, headache, and fever. And that these things that are signs and symptoms associated with meningitis, that meningitis can be caused by a whole slew of bacteria, several different fungi, and a whole lot of different viruses. So it can be caused by all those different things, but meningitis is meningitis, right? So that's the idea of what a syndrome is. It's defined by a complex of signs and symptoms. Yes. So in veterinary aspects, we would probably call that, that's a good question. How would I define that? Because sometimes dogs will limp as more of like a behavioral thing and not sometimes, right? And it's not always indicative of that. But I would consider it a sign because you can actually verify that it's happening. You're going to have a lot less going on symptom-wise with dogs because they can't tell you about things that you can't actually see and quantify, right? And so, yeah, sometimes they will, especially if you have Chihuahuas. They're yeah, right. So they can be, right. So that would be then at that point you would investigate as to whether or not you think this is due to some sort of physical thing that's going on with them, or if it's trauma. Because sometimes it's trauma, occasionally, especially with little dogs.
Over dramatic. They could just have a sticker.
Yeah. So my dog Meatball, has a tick. He's developed this thing where like when he runs outside, he lifts one of his legs up, and you'd think he was limping or something. It's just how he runs. And so that would be one of those things you'd have to, yeah, he just runs with one leg up. Like, I don't know. I imagine that when he probably had an injury and he just got used to running like that at some point, because he was a dog, he's a rescue dog. So I don't know, but these are good questions. When we start talking about the difference between signs and symptoms in the veterinary field, it's a lot more vague simply because you can't actually engage with the animal the same way that you can with people. Signs and symptoms a little bit more straightforward when you have humans, especially when they're conscious. When they're unconscious, that gets a little bit more tricky because there could be a lot more going on underneath that you can't actually get information from because they can't voice that for you. Here are some lists of these sorts of things that we would consider signs and symptoms if you wanted to go through this. So when we look at itching, someone might be scratching at their skin, but we still look at that itching aspect as being a symptom because we don't know if that's psychologically related. We don't know if there's something actually physically causing that need to itch. For example, I put those flea and tick drops on my dogs and it always makes them itch. Is it itchy because the feeling is different on their skin, or is it itchy because it's actually causing an allergic reaction, or is it itchy just like because mommy put drops on me and we hate when that happens and it makes us sticky? Like, I don't know. So that's a symptom. And you can't actually quantify how itchy it is. All right, so infections that are going unnoticed. This brings to this idea of asymptomatic and subclinical or inapparent infections. We know what asymptomatic would mean. You have an infection, you're not having signs and symptoms. Subclinical, it also kind of makes sense to us. You might have a slightly scratchy throat, but you're still infected. Like, you can have a scratchy throat without being infected, of course you can. But if you had a cold and you're getting over it, and you're recovering from that cold, and you've still got a slightly runny nose, that's subclinical, right? You still feel good enough to go to work. It's not impeding you from doing your daily life, but it's not enough for you to make you need to go to a doctor over. That's subclinical, right? We're not gonna talk about portals of exit. We're gonna move on with that. I need to move on to this. Latency, anytime that an infection hides out in the body. It could literally be hiding its DNA in your DNA, like we've talked about previously, or it could be like our tuberculosis bacteria. It gets into your macrophages, it gets eaten up by those guys, and it just stays there. And you could have the infection with tuberculosis without any symptoms for a very long time. We're talking decades of just having tuberculosis. And this is why we have you guys get tested for tuberculosis before you go work at hospitals, because you could literally just have it and not know. All right. So anything where you've been infected and you're carrying it around with you, but it's not actually causing disease presentation, that period is called latency. Yes. Yes, because they just want to double check and make sure that they didn't, because it can be unreliable. All right. Sequelae, these are effects of disease that are going to stay with you even after the disease has been cleared. So this would include, like when we see with polio, when, yes, did you have a question? Yeah. I was going to say, yeah. So when FDR was in the presidency, he was in his wheelchair. Did he have polio during that? No, he had polio previously. That polio made him paralyzed, and then he was in a wheelchair because of that. He didn't have polio anymore. And so that effect of that disease, even though it was cleared from his body, was still there. Those are sequelae. You can become deaf. We can have deafness from having meningitis, for example, that sort of stuff. So this leads us to our stages of infection, the course of infection. It lines up very well with our growth curve that we talked about with our bacteria, where we had the lag phase, the exponential or log phase, and then we had the stationary phase, and then we had our death phase. So this lines up pretty closely with that. In the lag phase, this is our incubation period. You don't have any signs or symptoms, because your body hasn't recognized that the thing is there yet, and it probably hasn't multiplied up. It hasn't really like established itself and started growing heavily with its infection. So this is that period of time from when you're first exposed to whenever you have your first symptoms. Okay, this can be weeks in some cases. It could be days to weeks. Then during the exponential phase, we kind of have two levels of our infection experience. First is the prodrome. The prodromal period is whenever you have your very earliest signs and symptoms. You know what this is, guys. You go home after working a shift at the hospital, and you're starting to get that sort of sore throat, that tickling in your throat, a little bit of drainage, and you're like, oh, it's coming, isn't it? I'm getting a cold or I'm getting the flu or something. That's the prodrome. It's the first signs and symptoms. Then later on in our exponential phase, that's the very, very beginning of your exponential phase. Then later on in the exponential phase, and as we lead into our stationary phase, we are going to be in our acute phase of the infection, when you're full blown. You've got all the symptoms of flu going on. Okay, you have the flu and you're feeling it. That's the acute phase. And then the death phase is when we see convalescence. Usually, you either get over an illness or you die. That's it. That's your option. That's your option. You either get over an illness or you die. So if you're getting over this illness, that period where you're having that decline in symptoms and you're starting to feel better, that's convalescence. All right, and then it has its own little chart associated with that. So where do our diseases come from? A reservoir is where these things exist naturally in their primary habitats. So when bats have Ebola, they don't have any signs or symptoms or anything, but they are harboring Ebola in their blood. Like koalas with chlamydia, yes. So they don't have any signs or symptoms of chlamydia, but they have an infection of chlamydia. So they have the infection, but they don't have the disease. These that exist naturally in the world and maintain these populations of pathogens are reservoirs. Wherever you got sick from, whether it was the bat that you hunted down in the forest and ate, or maybe it was like if you were hunting for bushmeat and you had a monkey that was sick. The monkey wasn't the reservoir, was it? The monkey was sick, but it was the source of where you may have gotten Ebola if you were hunting bushmeat, which is usually how a lot of the that's how a lot of Ebola infections start, is by hunting bushmeat in smaller tribes in Africa, and then they go get treated in the bigger cities and spread it on to folks there. A carrier is somebody that can carry an illness without obvious signs or symptoms. So this could be people who are asymptomatic, like we said, a third of people with COVID, or it could be people who are just gotten sick, but they don't have any signs or symptoms yet. They don't know, but they're still harboring this infection that's going in their body. And you can be contagious before you even have signs and symptoms. You can also be contagious when you're starting to feel better. We know this, right? You feel better, you know, you still have that cough a little bit, but you're feeling good enough to go back to work because you need to make money to pay your mortgage and pay for your kids. But you need to get back to work. You're still contagious. You know you're contagious, and you're probably going to be passing it on to other people. Those are our convalescent carriers. We have chronic carriers as well as our passive carriers. So transmitting these things like in a medical environment when you're not changing gloves or something like that. A few types of vectors. Vectors are things that can spread illness, but they themselves are not usually a reservoir or a carrier. So some of them sometimes can be. The places that we could get illness from people that are not people could include animals that are actually actively infected. They may not have signs or symptoms. Yes, so it could be like rabies. So it could be an example of that. But these guys are acting as vectors. So they're transmitting the disease to the human population, right? And so if you're actively infected with the disease, you're a biological vector. If you're come into contact with the disease, but you're not infected with it, like cockroaches and flies. They go land on some poo-poo and then come land on your food and you eat the food. The cockroaches and flies didn't get infected, but they were carrying that infection with them. They are mechanical vectors. Zoonoses are any diseases that come from animals to humans. This is an obvious one for rabies, right? So that would be an obvious one. So if it's maintained in the animal population and can come to people from animals where it's normally maintained, that is a zoonosis. So this includes flu, which is normally maintained in avian or swine populations, and then can get passed on into humans. There's some other examples here as well. Communicable disease is anything that can be transmitted from person to person. A contagious disease is highly communicable, which means more easily spread. Non-communicable is a disease that you get that could be an infection, but you're not going to spread to somebody else. If I get a UTI, am I going to spread that to you guys? No, that's non-communicable. Horizontal transmission, getting it from another person. Vertical transmission, getting it from usually the mother, but it can be inherited from a father side through the sperm. Okay. If it's transmitted from the mother or father because of the birthing process, vertical. If it's transmitted from anything else, anybody else, it's horizontal. Oh my gosh, I'm not gonna. Mother or father, like the birthing or incubation process, pregnancy, things like that. All right, so indirect transmission, means that the thing that you're getting it transmitted from was not infected. So if I cough all over this pen and then I hand it to one of you guys and you get COVID from me, I don't have it, just to be clear. But if I did, and you got the COVID from me, is the pen infected? No, it's contaminated, right? But this will be a vehicle. In particular, we call it a fomite, an inanimate object that's not actually harboring that infection, but it's temporarily being used to transmit the infection. Now, another example of a vehicle would be if I had a water pump up here and I like vomited into it with my, I don't know, GI illness, and you guys were continually getting water out of it and you didn't know I vomited into it. That's a vehicle, but it's not really a fomite because it's maintaining that infection in there, okay? Oral fecal, that would be an example, probably oral fecal, but anytime that's coming from the body, usually you're pooping, you're not washing your hands enough. It's the best example, right? Before you're going and handling food, you should be washing your hands if you've gone to the bathroom. That's the best example. You can get it from water and air. If you're coughing or sneezing, you're releasing droplets into the air that can contain our pathogens as well. Robert Koch came up with some steps to help identify our microbes, basically by isolating them from the microbes, putting them into a new organism, and then seeing the same disease and then re-isolating that organism back out. That's Koch's postulates. All right, I'm gonna go over the questions really quick. Disease is any deviation from health, usually from an infection. An endogenous infection came from the body originally. A headache, is it a sign or a symptom? It is a symptom. All right. And if y'all think we're done, forget it. We have a whole other chapter. I want to at least touch on some topics from before we move on. All right. Epidemiology, definitely this chapter for chapter 14 is a flashcard chapter. Please take the time to make your flashcards and learn the terms from this. Epidemiology is basically studying the spread of disease. Look into what Florence Nightingale and John Snow did for us. Florence Nightingale is the mother of nursing. She's well known as the mother of nursing. And what she did was basically identify people who were dying in a hospital, realize that these people didn't have GI symptoms before they came to the hospital, and now they have dysentery. Why is that? Doctors weren't washing their hands. She started having people wash their hands. People got less sick. John Snow identified a cholera epidemic, basically from a water pump, that people were spreading cholera through that water pump, and they kept using it even though he told them to stop. He took the handle off. Guess what stopped spreading? Cholera. It's the father of epidemiology. All right, moving on. Prevalence, incidence, morbidity, and mortality. Know the difference between these terms. Prevalence, the total number of cases in the entire population of a disease. Incidence, the number we had this month. So when we come out with hospitals or the CDC or something like that, talking about weekly morbidity and mortality, morbidity is the same thing as incidence. So weekly, how many cases did we have this week? How many cases did we have this month? All right, that's the same thing as morbidity. Mortality is death rates. Okay. Point source, common source, and propagated epidemics. Point source, it came from one point at one time. I have COVID, I came in here, I coughed and sneezed all over you guys, and you got COVID. You know exactly what point in time and what place you got it from. Point source epidemic. Common source epidemic, the water pump in London with the cholera in it. People were coming to that water pump over a vast wide of time, right? They were coming over there over weeks, and people continuously getting cholera. It was the common source, but it didn't happen at a single point in time. Okay, common source epidemic. Whereas propagated epidemic, like COVID, we're maintaining COVID in our population now, aren't we? It comes and goes in waves, but it's still maintained in the human population. That's a propagated epidemic. Next, we have index case, the first case, endemic. These are diseases that are naturally found in the population. Would you be surprised today if you wound up randomly with a common cold? No. But if everybody was getting the common cold, like let's say everybody in this class got a common cold and everybody in physiology and everybody in all these other classes, let's say the whole campus got a common cold. We'd be pretty freaking surprised then, right? Then it's a problem. So endemic is having that disease at this current, basically, incidence, okay? Having it there at this relative steady frequency. Sporadic disease. If I got bitten by a coyote and got rabies today, that's sporadic. It's not really going to spread around and cause an epidemic, right? But I did get it. That's called sporadic. And it's not endemic either, right? We're not used to seeing it. Epidemic. Now it's exceeding what's endemic. So we had just a shitload of people coming down with a cold, or like a whole bunch of people got RSV. You might expect to see a few cases, but just all of these people now getting it, that's an epidemic. Once it exceeds what is endemic. If we had 10 people come in here with Ebola, that would be an epidemic because it's exceeded what we normally see in our population, right? Pandemic, anytime it spreads across continents. We have screening tests that we can use. Screening tests are usually cheaper, but less effective. So they are not as sensitive. They're not as selective, but they're a quick, easy, effective way to just diagnose if you think somebody has a disease or not. So this includes a pap smear. We're not actually trying to diagnose you if you have cancer or not, but whether or not we think it's likely that you might have it. So that's a screening test. Tests can be looked at as specific or sensitive. We want them to be both. Sensitivity deals with if you're a sensitive person and somebody's not a sensitive person. The sensitive person picks up on every little cue from everybody, right? The person who's not sensitive won't really see it. So if you're a sensitive test, you're picking up on all of the positives, even the little weak ones. You see all of the positives. The more sensitive you are, the more positives you see. Okay. So then you're less likely to have false negatives. You're less likely to call something negative when it's not negative, right? Specificity, this is reducing false positives. You're less likely to call something positive that's not positive. This might look like this person having a runny nose and a cough, but this person's having aches and pains and a fever. This person's not having aches and pains and a fever. You don't want to call them both positive for the flu when this person's not even having aches and pains and a fever. So specificity, being sure you're calling what's positive is actually positive. You want, of course, to be only calling positives positive and negatives negative, and then therefore that's specificity and sensitivity. Okay, there's an audio short on this. If you get confused, I'll explain it in more detail. So if you're trying to look at the cause of disease, you can do these tests in order to look at that. In epidemiology, there's 3 major tests. The first one is cross-sectional. You're taking a single point in time and looking at population of people in that single point in time and whether they have disease or how they were exposed or something like that. You're not necessarily looking for people that have disease versus people that don't have disease. We're taking a single point in time and all the people in that point at that area and looking at all the data that you, usually 2 points of data that you're looking at there. Whereas case control, so cross-sectional, looking at points of data in that 1 point in time. Whereas case study, case control, you're taking cases, people that have the disease, and control, people that don't, and comparing them and trying to delineate what the difference is between them. Looking at aspects of people that have it versus people that don't have it, and then trying to break down what the difference is. Instead of taking at 1 point in time, which is cross-sectional, collecting data, all relating at that 1 point. Cohort studies are longer term studies. These are examples that would include taking somebody that smokes cigarettes. I smoke cigarettes, just to be clear, and don't have lung cancer. I don't have lung cancer yet, right? So taking somebody that smokes cigarettes that doesn't have lung cancer and somebody that doesn't smoke cigarettes that doesn't have lung cancer and watching them over like 10 to 20 years and seeing which population is more likely to have lung cancer and then being able to correlate that saying, yes, if you smoke, you're more likely to have lung cancer. That's a cohort study. Make sense? There are reportable diseases, and if you work in healthcare, there are certain diseases that they are going to keep track of, including catheter-associated urinary tract infections, extremely common because it's really hard to sterilize that area, and the equipment that they're using is often reusable. So making sure that is sterile and stays sterile is like next to impossible. Central line infections. These are also same sort of thing. It's just hard to keep the area completely sterile. And usually you have a central line for a very long time. So it's just hard to keep it clean. Surgical site infections. You're often relying on the patient for self-care. And people are bad at that. Ventilator associated events. Ventilation and having just a constant source of introducing new bacteria into this environment. And if people aren't coughing as much, they're usually not conscious or they're unable to control coughing as well. They're not sitting up, they're laying down. Fluids will basically sit in the lungs and lead to increased infection rates. Usually have an infection control officer at hospitals that are controlling that. Some ways that we maintain cleanliness at hospitals is through medical asepsis or surgical asepsis. Medical asepsis, washing your hands and changing PPE. Surgical asepsis, you want to try to keep every single thing in your environment sterile as possible. Not as clean as possible, sterile as possible. So it's completely devoid of any life whatsoever. So we're not relying on just washing hands here. We're trying to sterilize everything that goes into that room, right? So there's a difference between those. Are not, this is the number of people you're likely to infect if you have a disease. For example, with COVID, I think the Delta version, it might be Omicron, I don't remember which one. whichever one was the most infectious through Omicron. Anyways, the most recent one that we had, if you get COVID, you're likely to infect 5 other people. That's the R naught for that disease. If you get a disease, how many people are you likely to infect on average? That's the R naught. The case fatality rate. This is when you get the disease, the likelihood that you're going to die from it. Not the likelihood that everybody's going to die from it. The likelihood that if you get the disease, that you're going to die from it without treatment. So HIV, the likelihood that you're going to die from it with treatment is completely different than the likelihood you're going to die without treatment. HIV is 100% fatal without treatment. But if you get treatment, you can maintain a perfectly normal life and die of natural causes, right? So case fatality rate deals with no treatment. All right, so there's some charts here that look at the deadliness, the case fatality rate versus R naught for some diseases. And a lot of times they're inverted because as it turns out, diseases that kill people really easily just aren't as transmissible. Usually you aren't going to hang out with somebody that's dying that much, try to get contaminated from them. We just don't see it spread quite that way. And then herd immunity. Vaccine plays a huge role in herd immunity. Herd immunity basically says that for each disease, there's a certain population of people that need to be vaccinated in order to protect the rest of the population. So for measles, in case you don't know, we're having an outbreak of measles here in Oklahoma, not just in anywhere. I'm talking about here in Oklahoma, we're having an outbreak of measles right now because of anti-vaccine. In my class, we are pro-vaccine as long as you're in my classroom, okay? And if you're going into the healthcare world, thoughts and prayers with you. If that's all I'm going to say. So herd immunity is a real thing. 95 to 98% of people have to be vaccinated for measles to protect the people that can't get vaccinated. I'm not talking about people that choose not to be vaccinated. I'm talking about people that can't. We're talking about little babies that can't develop their own immunity yet, or old people whose immune systems are failing them. They cannot get vaccines. So you choosing not to get the vaccine because you don't believe in how vaccines affect you, or you think they're just a gimmick from pharmaceutical companies or whatever. The outbreak of measles here in Oklahoma proves that the failure of vaccines will reintroduce these diseases back into our communities. It will, and it is. You're not getting your vaccines, have fun with measles. You think measles isn't a big deal? Wait till your child dies from it. You'll think it's a pretty big deal then. And if my little baby died, my little infant baby that couldn't get its vaccines died because you decided to just, you know, fuck the system and whatever, and you're not going to get vaccines, I'm coming for you. So I'm just saying, you can write it down, folks, because, yeah. Yes, you only get vaccinated as kids. They can do titers for you. So if you're going to nursing, they actually take your titers to make sure your titers are high enough. And if they're not high enough, you don't have high enough antibodies, you have to get revaccinated for measles. So I actually had a student of mine from last semester, her titers were low for measles. She had to get her whole new series of MMR vaccines done. So, yeah. Possibly not. Yeah. Your health insurance may pay for you to get revaccinated or it may not. So it just depends, right, on your, good luck with that too, right? Aren't we? Oh, I just threw it. So let's go up to the lab, guys. We got to get out of here. I think that was the end, right? We're close enough to it. Bioterrorism and stuff. I know that was a lot. You guys did great.